Well, hello everybody. We're going to keep on keeping on here, finish up chapter 13. This is part 2 of chapter 13, just 5 pages from the textbook, pages 465 to 470. What we're going to talk about today is the regulation of metabolism. We'll frame it first conceptually, why metabolism even needs to be regulated. That'll bring us to catabolic pathways briefly to sum up what we talked about last time. And anabolic pathways, making things, really the first true anabolic pathways we'll discuss in this course will happen today. Talk a little bit about feedback regulation and how that pertains to glucose metabolism, how cells, both animal and plant cells, store excess glucose as energy. And like I mentioned at the end of the last video, this one is going to be a short one, probably quite short. I have to say the last video wasn't as long as it felt, as dense. I know a lot of information, but all the same, this will be a short lecture. Just in my opinion, there was no logical breakpoint in the last lecture. That's why I crammed so much information into it. The glucose metabolism really is one long flowing story, and it didn't seem right to break that story up before the end of the citric acid cycle. But again, having a long video, or a dense video, I should say, last time allows us to have a little bit of a lighter video today before we move on to the electron transport chain and oxphos next week. So let's do it. Let's talk about the regulation of metabolism. Cells strive for homeostasis, and as we've talked about in this course so far, that doesn't necessarily mean equilibrium. In fact, cells strive to stay away from equilibrium, but it just means having all different cellular processes in balance. And when I say that, I really mean all cellular processes. I mean all of metabolism. This enormous network of reactions that all intersect, crosstalk, and feed into each other have to be finely and precisely tuned so that the cell is using its limited resources and its energy in the best way possible. And for that to be possible, for all of these reactions to be coordinated, to have efficient use of energy, to run smoothly together, to not oppose one another, to respond appropriately to signals that are coming in both from outside the cell and inside the cell, we need regulation. We have to have means by which these reactions can be turned up or on or down or off so that all of this coordination and fine-tuning can be possible. So enzymes have to be turn-onable or turn-offable as needed. The precursors for these different enzymatic pathways need to be available and need to be shuttled or shunted to other reactions if they're in excess. Intermediates and metabolites all need to be sent to the pathways and reactions where they could be most efficiently used and taken away from reactions where they're redundant or wasteful. In addition to that, though, cells must have ATP levels as an absolute priority. They have to make sure that the paramount priority of the cell is having ample energy on hand, because without that, the cells will die. That said, in the normal course of our lives, in fact, in the normal course of our day, we have ATP deficiencies. We sleep and go long periods of time of fasting simply because we're not awake. Seasons change, which, at least for the bulk of our evolution, led to a much shorter supply of food in the winter. Things we're hunting can get away. We can have long streaks of bad luck as we're trying to hunt our food. Drought kills plants, etc. So we can't always rely on a ready source of food. And this is all artificial now, right? The context now is that we just go down the street with a couple bucks and we go to the supermarket or the McDonald's and get food. But that is not natural, and that's not what we were evolved for. That's a uniquely human circumstance. Life is supposed to struggle a bit to get its food, and certainly that's the context in which we evolved. So because of this, cells require some mechanism for energy efficiency and for energy storage. When we have plentiful, bountiful times, food is more abundant than we need it to be, it would be wasteful to not eat that food when it's around. And so we have evolved mechanisms to store excess food as excess energy for times when it might be depleted. And you can see in this diagram a lot of things that we're already familiar with. Glucose becomes glucose 6-phosphate, becomes fructose 6-phosphate, which becomes fructose 1,6-bisphosphate. These are the first 3 steps of glycolysis. But now we see an offshoot. We see a side reaction. If glucose 6-phosphate levels are high enough, it is mutased, right? We move that functional phosphate group. It is mutased to glucose 1-phosphate, and glucose 1-phosphate is a precursor for glycogen synthesis. Glycogen is how our cells store excess sugar, excess energy. But this implies another level of regulation. It's not enough anymore to just regulate these individual steps of glycolysis. We need an additional sensor or additional level of regulation that can detect when glucose 6 phosphate levels are in excess and then shunt that excess glucose 6 phosphate to glycogen synthesis. Glycogen synthesis, not surprisingly, needs its own enzymes, which are also regulated, and that's a lot of what we'll get into today. So when sugar and other energy sources are plentiful, we've evolved these intersecting and cross-talking pathways so that we can eat to excess. We have an urge to eat to excess, and then we can take those excess monomers and not metabolize them for ATP immediately, but store them on board in our bodies as excess energy that we can then tap into and release later on when food isn't so plentiful. And just as a brief digression, this is why sweet things strike us or are perceived in the way that we perceive them. Sugar, which is what we're talking about here, is the most efficient molecule to metabolize. It is easier for us to get energy out of sugar than anything else. And sugar is actually quite easy to store too in this glycogen form. So what happens when we're full, we're sated, we're no longer hungry, and then something sweet and sugar rich is put in front of us? We continue to have an appetite for that. We can eat to discomfort, sweet after savory. And the reason for that is because of this evolutionary pressure. Sugar is so rare in the natural world and so precious in terms of its energy potential that when something sweet makes itself available for us, we're hungry for it, even if we're not hungry. I should say we have an appetite for that sweet thing, even if we are not truly hungry. This is the nature of dessert. This is where dessert came from. Get a big steak, baked potato, and a whole plate of vegetables at the restaurant. You're full, you're uncomfortably full, maybe you're loosening your belt. Oh my God, that was a great meal. I'm going to have to roll me into the car to get home. And the waiter or waitress comes over and says, "Would you like to see the dessert menu?" Well, now that you mention it, I would. Can I please have a slice of chocolate cake with ice cream on top? The reason why we can eat that after a full meal is because of this evolutionary pressure to eat sugar when it's available, because that excess sugar can be stored for when food isn't so plentiful. The only problem with our culture and with our evolution at this point is that for many of us, for most of us at least in this culture and in this country, there really isn't time where food is not available. And so we eat to excess without needing to ever get to those stores. So when to oxidize a sugar for immediate energy release and ATP production, and when to store it for later when food might be less plentiful, is another gateway of regulation, is another thing that needs to be monitored and regulated. It's all enzymatic. So, not surprisingly at this point, metabolic reactions are organized and they're precisely regulated. When we talked about glycolysis in the citric acid cycle in the last video, those were 18 reactions we discussed. 10 reactions in glycolysis and 8 reactions in the citric acid cycle. 18 novel enzymes responsible all to get us all the way to reduced NAD and FADH2. Part of why it takes so many reactions to do this, we've already discussed. You want to release the potential energy of those covalent bonds in glucose a little bit at a time, these small negative delta Gs that, one, allow you to capture that energy as reduced electron carriers or triphosphates, and two, avoid this explosive release of energy that might fry the cell. But another side benefit of having so many reactions and so many enzymes involved is that it gives us multiple potential points of regulation. Each one of those 18 enzymes could potentially be regulated and allow us to very precisely and finely control the pace and the abundance of these reactions occurring, and also allow us to shuttle off or shunt off those intermediates as they're made. So those intermediates, they can certainly become electrons, no doubt about it, but they can also become a lot of other things too. The intermediates of glucose metabolism have many different roles in the cell, and we touched on that in the last video a little bit when I showed that enormously complex, almost graduate level metabolic diagram. Pyruvate is a perfect example. Pyruvate is a reactant or a substrate for more than half a dozen different biochemical reactions, which each use pyruvate for a different purpose. Now, I know that pyruvate is the product of glycolysis, but that's not what I'm talking about here. Pyruvate is the starting point for many other anabolic reactions. And here are some examples. Yes, pyruvate can become acetyl-CoA and then be entered into the citric acid cycle, can combine with oxaloacetate and become citrate. This is the pathway of pyruvate we've already discussed. But pyruvate can also be converted into lactate. Not so much because cells need lactate for anything, but that's the fermentation pathway. That's how our muscles can continue surviving. I almost said thriving, but that's not true. That's how our muscle cells can continue surviving in a short-term absence of oxygen. We just have to recycle NADH to NAD+. We talked about that too. But pyruvate can also be converted into acetaldehyde. Acetaldehyde is used in many different biochemical reactions. Pyruvate can be converted back into phosphoenol pyruvate to kind of do glycolysis in reverse. We'll talk about that in a second. Pyruvate can be immediately converted into the amino acid alanine by adding an amino group to it, adding ammonia to it, and then alanine serves as a precursor for making many other amino acids. So pyruvate is the starting molecule of amino acid biosynthesis. And check this out. If you carboxylate pyruvate, if you add a carbon dioxide to it, pyruvate becomes oxaloacetate. So if you want to have more citric acid cycles, you can convert pyruvate into oxaloacetate and a new citric acid cycle is born. So pyruvate can become acetyl through decarboxylation and be fed into the citric acid cycle, or pyruvate can be made into oxaloacetate by carboxylation and become a new citric acid cycle, which is crazy. And that's just one example. Other intermediates that we talked about in the last video have similar multimodal uses throughout the cell and can become many, many different things. So one of the take-home messages of this is, if ATP levels are already high, why are you doing glucose metabolism? Why would you convert pyruvate to acetyl-CoA to harvest electrons that can only be used by the ETC to make more ATP if ATP levels are already high? Instead, if ATP levels are high, use that pyruvate for something else. Use it to make more citric acid cycles. Use it to make amino acids. Don't use it to make more ATP if ATP levels are already high. But if we're implying a gateway of decision, should I use this pyruvate for respiration or should I use this pyruvate for something else? That requires regulation. That's what regulation is. It's a cellular decision point. And the general flavors or categories of regulation that are involved in making this decision are really not new to us. They use strategies we've already talked about. Some of these enzymes that are regulated by high ATP levels or high pyruvate levels are allosterically regulated. Well, we know what that means. Or they're regulated by reversible covalent modification. That's just a long way of saying phosphorylation. They're regulated by kinases and phosphatases. They can be regulated by being broken down completely, destroyed on demand. The proteasome does that. They can be regulated by feedback regulation, which we've already discussed. In fact, the only thing we haven't broached yet on this list is the regulation by isozymes. But all these other strategies are at least somewhat familiar to us already. So let's start talking about it. Let's start talking about how these kinds of enzymes are regulated to achieve these offshoots, these other reaction pathways that allow the cell to be efficient and make the best use of its available molecules. Feedback inhibition. Again, glucose is critical. For muscle function, we require glucose. That includes breathing, the muscle contractions of the diaphragm. The brain, the only thing the brain can use for energy is glucose. The brain can't metabolize amino acids like most other cells can, and the brain can't metabolize fats either. So for the brain to continue functioning, we need glucose. So essentially, any long-term deprivation of glucose will result in cellular death, organism death. We need glucose to survive. When glucose is needed, but we don't have any stored, and we'll talk about glucose storage in just a second. So when glucose is needed for, let's say, brain function, and glucose stores in the body have been completely tapped out, the only real mechanism we have to keep the brain alive and to keep glucose present is a process called gluconeogenesis. And that translates to creating or making, that's genesis, new, neo means new, glucose. So gluconeogenesis means making new glucose, making glucose from scratch. And this process of gluconeogenesis can be used to make glucose, glucose that can then be respired to create ATP and something like the brain continues to survive. So essentially, and pretty much literally for the most part, gluconeogenesis is a reversal of glycolysis. 7 of the 10 reactions of glycolysis can occur just as easily in reverse as they can occur forward in the direction that we talked about in the last video. So it just depends on the concentration of reactant and product, really. That skews the equilibrium in one direction or not. So most of the reactions of glycolysis are directly reversed to make gluconeogenesis possible. And you can make 1 glucose from 2 pyruvates. However, there are 3 steps, steps 1, 3, and 10, that are irreversible, meaning the enzymes only move in the forward direction for glycolysis. And so to reverse these steps, you need different enzymes. You need a unique enzyme to do step 10, step 3, and step 1 in reverse. And what we see here on the screen is step 3, you might remember it. Fructose 6-phosphate is being phosphorylated to fructose 1,6-bisphosphate by our old friend PFK, phosphofructokinase. That's step 3 in glycolysis. The reverse of this step for gluconeogenesis to be possible is the dephosphorylation of fructose 1,6-bisphosphate to leave us with fructose 6-phosphate, remove the phosphate group. But that requires a completely different enzyme, fructose 1,6-bisphosphatase, because remember, phosphatases remove phosphate groups. So how do cells know which enzyme to be using? How do cells know when to go in the forward direction with PFK, so that glycolysis would occur, versus how to go in the reverse direction with fructose 1,6-bisphosphatase for gluconeogenesis? And the short answer is regulation. But the longer answer is that that decision is made at those same 3 irreversible steps. Step 1, step 3, and step 10 all determine which direction this pathway goes into. If the enzymes for glycolysis are activated through regulation, and along with that, the enzymes for gluconeogenesis are downregulated, then you're going to do glycolysis. You're going to make pyruvate. If vice versa, the enzymes that catalyze step 1, 3, and 10 are deactivated through regulation, and the reverse enzymes are turned on, well, then you're going to be doing gluconeogenesis, and you're going to be making glucose. Phosphofructokinase, PFK, again, the enzyme that catalyzes step 3 of glycolysis, it phosphorylates fructose 6-phosphate to fructose 1,6-bisphosphate. In other words, what fits into its active site is a sugar, is fructose. Therefore, to be sensitive to ATP levels, PFK can't have ATP bind to its active site. It needs to be allosteric. PFK needs to have its sensing molecule, its detector molecule, ATP, because that's the levels we're interested in, bind to some secondary site elsewhere on the enzyme, because it won't fit. ATP is not going to fit into an active site that is optimized for a sugar. So PFK is allosterically regulated, just as we've talked about before, and it's allosterically regulated by the end product of its net reaction. That's feedback inhibition. What are we trying to make here? ATP. What's going to regulate PFK? ATP. So again, since the substrate of this enzyme, PFK, is a phosphorylated sugar, and we want this sugar to be, this enzyme to be sensitive to a nucleotide, to ATP, ATP is not going to fit in that active site. In order for ATP and then the dephosphorylated forms of that, adenosine diphosphate and adenosine monophosphate, in order for those to bind to PFK and regulate it, they have to be binding to an allosteric site. It's the only way we could possibly have these nucleotides bind to the enzyme. And they do. And they regulate PFK in a very intuitive and logical way. First and most importantly, when ATP levels are high, we see inhibition of the enzyme. Well, that makes sense. Why is PFK turning fructose 6-phosphate into fructose 1,6-bisphosphate? In order to make pyruvate, in order to make acetyl-CoA, in order to run the citric acid cycle, in order to harvest enzymes, in order to make ATP. And so again, if ATP levels are high, we want PFK off so we don't end up making more ATP. ADP and AMP have the opposite effects. When their levels are high, PFK is activated. Well, let's think about that. If adenosine diphosphate or adenosine monophosphate are high, that means the cell has hydrolyzed much of its existing ATP and burned that ATP already for energy. In other words, high levels of ADP and AMP are actually indicative of low levels of ATP. If ATP levels are low, we need to phosphorylate more ADP to make ATP. That means we need fructose 1,6-bisphosphate to make pyruvate, to make acetyl, to harvest electrons, to make ATP. And then I love this citrate one, even though it's not really part of what the textbook mentions. If citrate levels are high, we inhibit PFK. Well, we got to think about that one. I'm strumming my chin here. If citrate levels are high, that means a lot of oxaloacetate has been converted into citrate. Well, the only way that's possible is if there was a lot of acetyl-CoA around. So high levels of citrate are essentially they're indicative to the cell that electrons are on the way. The citric acid cycle is cranking at full throttle, and we're going to get a lot of electrons that we can use to eventually make ATP. So if ATP is in the process of being made, let's store that excess sugar for another time, and PFK is inhibited. So very logical regulation mechanisms when we think about what the cell is trying to do and how the cell is using the information from these molecules to tell itself about its energetics. So essentially, ATP levels when they're high turn PFK off, good, and the other dephosphorylated forms of ATP, ADP, and AMP turn PFK on because that means ATP levels must be low. It just makes sense. It makes logical sense if you understand where the molecules came from and what information those molecules are telling the enzyme, telling the cell. Surprisingly, gluconeogenesis is energetically costly. It takes 4 ATPs to make gluconeogenesis, so making glucose from pyruvate burns a considerable amount of energy. But from the cell's perspective, it's worth it when gluconeogenesis is needed because the brain can't use fats or proteins for energy. So if we're in this state where gluconeogenesis is needed at all, then we're already in fairly dire straits. We can afford to burn some ATP to make the glucose that the cell needs in order to function. And fats and amino acids can be used to make ATP by other cells. So the idea here is gluconeogenesis often happens in the liver. Let's have the liver break down fats and amino acids to make ATP, and let's invest some of that ATP to making glucose that the bloodstream can then bring to the brain, and the brain continues to function. That said, gluconeogenesis only makes logical sense for the organism if there's no other possible source of glucose available. This includes, of course, consumed glucose, but it also includes glucose stored forms of glucose. And we do store glucose. Animal cells, including our own, store excess glucose as a molecule called glycogen. And glycogen is stored by the liver and by the muscles. Interestingly, muscle cells store glycogen selfishly. They store glycogen for themselves and themselves only. The glycogen stored by muscle is broken down by muscle and respired by muscle to make ATP for muscle. Liver cells store excess glucose as glycogen selflessly. They store it on behalf of the rest of the body, mostly the brain. And so when system-wide levels of glucose go low, it's the glycogen from the liver cells that's broken down and provides that glucose to the body, but to the brain when they get low as well. And glycogen is a branched polysaccharide. It is just glucose monomers stuck together in long branched chains. And then these chains form nearly insoluble granules that you can actually see in the cytoplasm of liver and muscle cells. That's what we see there on the screen. So when ATP levels are low and glucose levels are low, those are going to be signals to the cell that it's time to break down glycogen. And glycogen will be broken down in muscle for the muscle. Glycogen will be broken down in the liver for the brain and the rest of the body. And when glycogen is broken down, the glucose monomers are popped off one at a time off these branched chains and then metabolized. It's a phosphate that actually breaks the bond that holds the glucose monomers together. And so when glucose is broken off of glycogen, it's broken off as glucose 1-phosphate. There's a phosphate group on the first carbon. And you might remember, I hope you remember, from our glycolysis video last, that the product of the first reaction of glycolysis, the second precursor in glycolysis, is glucose 6-phosphate. So what happens is when glucose 1-phosphate is released from glycogen, a mutase enzyme gets involved, moves that phosphate from carbon 1 to carbon 6, converting glucose 1-phosphate into glucose 6-phosphate, and that glucose 6-phosphate enters right into step 2 of glycolysis, becomes fructose 6-phosphate, fructose 1,6-bisphosphate, and off to the races we go. Not surprisingly, I would think at this point, glycogen synthesis, making glycogen when sugars are in excess, and glycogen breakdown, releasing glucose from glycogen when sugar levels are low, all tightly regulated, and tightly regulated by feedback inhibition, where the products of these processes serve as signaling molecules to allow the cell to detect its needs. Interestingly, though, glucose 6-phosphate regulates both enzymes of glycogen, but in opposite ways. And by both enzymes, I mean the enzyme required for glycogen synthesis, which is called glycogen synthetase, and the enzyme required for glycogen breakdown, which is called glycogen phosphorylase. Those two enzymes are both regulated by glucose 6-phosphate. It's just the effect of glucose 6-phosphate is opposite for each of them. When glucose 6-phosphate levels are high, meaning we have a lot of glucose in the cell, we have glucose excess, high levels of glucose 6-phosphate activate glycogen synthase. So glycogen synthase is the enzyme that takes individual glucose monomers and links them onto glycogen. So when glucose 6-phosphate is high, meaning we've got more of it than we need, a mutase comes in and moves that phosphate to carbon 1, and then glucose 1-phosphate is linked onto a glycogen tree and stored for later. Low levels of glucose 6-phosphate activate the enzyme for glycogen phosphorylase. This is the enzyme that breaks off these glucose monomers and releases glucose 1-phosphate. So high levels of glucose 6-phosphate turn an enzyme on. Low levels of glucose 6-phosphate turn an enzyme on. But the enzyme that's turned on by high levels of glucose 6-phosphate is the glycogen-making enzyme. And the enzyme that's activated by low levels of glucose 6-phosphate is the glycogen breaking down enzyme. So here we have it in words. Glycogen synthase, the enzyme that makes glycogen, is activated by glucose 6-phosphate if its levels are high. And glycogen phosphorylase is activated by low levels of glucose 6-phosphate, or to say it the other way, is inhibited by high levels of glucose 6-phosphate. If glucose 6-phosphate levels are already high, why in the world would we break down glycogen? Let's allow glycogen synthase to make more glycogen stores because we have plenty of sugar on hand. And ATP also inhibits glycogen phosphorylase. For the same reason. If we have plenty of energy, let's store that excess sugar for later. Now back to fats. Remember, fats, and by fats we mean true fats, long-term energy storage fats, are triacylglycerols. These are glycerol with 3 acyl groups, 3 fatty acids coming off of them. This is our long-term seasonal hibernation energy storage. On average, humans store about 1 day's worth of energy in glycogen and 1 month's worth of energy in fat. That's a healthy, typical human being. Triacylglycerol droplets can be seen as stored forms inside individual cells, and adipocytes, said it correctly, adipocytes are cells that make up adipose tissue, and these are specialized cells that exist solely for storing fat. What you see here in blue are the actual cells, and what you see in red, deep red, are the adipose granules, the droplets of fats that are in these cells. And so adipocytes exist solely to make as much fat droplets in them as they can, and they exist as storage cells for fats. That said, despite fats being long-term storage, it's not as though we only dip into our fat stores in the winter. We dip into our fat stores anytime we have a prolonged period of fasting, and that includes overnight fasting while sleeping. We burn through quite a bit of glycogen while we're sleeping, and so the cells will revert to beta oxidation and begin to break down fats while we're sleeping as well. In fact, most of the acetyl-CoA that enters the citric acid cycle within 1 hour of us waking up is coming from fatty acid breakdown instead of glycogen breakdown. So what's keeping us running first thing in the morning is fat metabolism. And then we eat breakfast or we have a sugary coffee, we consume some form of glucose, and we revert from fat breakdown to going back to respiration and making the ATP from the glucose we just consumed. So yeah, we've been at it for about half an hour and we're going to start wrapping up here. Let's wrap up not by talking about our own cells, but by talking about some of the cells that we consume for energy, specifically plants. Now, plants respire much of the sugar they make by photosynthesis, just like we respire sugars. So plants will absorb sunlight, take in water, and absorb carbon dioxide from the atmosphere. They'll use that sunlight energy to build bonds between the carbons of the carbon dioxide, and then they'll hydrate those carbons with the water they consumed, making carbohydrates. So plants take water, carbon dioxide, and sunlight and use it to make glucose. And then the mitochondria in those plant cells respire that glucose just like we do and make ATP from it. So the plants are eating, respiring, the sugars that they create by photosynthesis. However, plants have gotten very good at this, and there are many, many days where carbon dioxide, sunlight, and water is plentiful. And when that's the case, those plant cells make more glucose than they need. And they store that glucose very much in the same way we do, except they don't make glycogen, they make what we call starch. Starch is actually a combination of two different closely related polysaccharides of glucose. One is a coiled helix called amylose, and the other is a branched tree that looks much like glycogen, but is different in its bonds and some of its chemistry. But superficially, amylopectin and glycogen look very, very similar. And plants also make fats as triacylglycerols, just like we do. Think an avocado, right? Avocado oil, olive oil. These oils are fats. They're plant fats. Fats that the plant has made with excess energy that it made through photosynthesis but didn't need. And where the plants often put a lot of this starch and fat is in, not surprisingly, its offspring. Seeds are nothing more than a plant embryo surrounded by an enormously rich and plentiful food source. If you ever break a seed open, whether it's a pea seed, a nut of some kind, a lima bean, you've got a little tiny, almost looks like a leaf in there. That's the embryo of the plant. And the rest of the body of the seed is food. It's starch and fat, there to nourish that plant until the plant grows tall enough and sturdy enough to break through the soil and begin doing photosynthesis for itself and survive in a self-sustaining way. In plants, these fats and these starches are stored directly in the chloroplasts where the glucose is made by photosynthesis, which is both efficient and logical. The glucose is first made in the chloroplast. When the glucose is in excess, it stays in the chloroplast and is used to make starch or offshot to make fats with the excess hydrocarbons that glucose offers. And this is why seeds and nuts are so good for us in terms of health, because those fats tend to be unsaturated fats. They don't clog the arteries as much, and good for us in the terms of tasty. I'm currently and ashamedly in the middle of a macadamia nut kick. Can't get enough of them. I have a major issue with Doritos. I can't open a bag of Doritos. Someone has to open it for me and portion it out for me like I'm a 3-year-old, because otherwise I can't be trusted with the bag. But even despite that major weakness, macadamia nuts is giving Doritos a run for their money. The other day I reached into the cabinet and actually chose to grab a bag of macadamias instead of Doritos. Why? Because evolutionarily, physiologically, I know that those macadamia nuts are rich in fats and starches. Sugars and fats is what that seed is. And the more of seed there is, the more fats and sugars there are. And again, that's what we're evolved to eat. That's what we want to eat when it's available, because we can store those for later when we need them. So I should have switched the slide a minute ago. I apologize, but this is an electron micrograph of a chloroplast, and you can see very large starch granules and fat droplets in that chloroplast, in the organelle of the chloroplast, there for the cells to store later when energy is plentiful. So yeah, it looks like 36 minutes, and that's pretty much all I've got for you. So to summarize what we talked about today, first, metabolism is very tightly regulated to give the cells precise control over their entire internal physiology. That's what homeostasis is, and allow the cells to make the most efficient use of the energy that they have on hand. Whenever possible, excess energy is stored for either short-term sugar storage as glycogen in an animal cell, or long-term energy storage as triacylglycerides or fats in an animal or plant cell. And then we can use those stored forms of energy during energy shortages, during periods of fasting or starvation. Glycolysis takes glucose and makes 2-pyruvates with it. Gluconeogenesis takes 2-pyruvates and makes a glucose with it, making gluconeogenesis a reciprocal pathway of glycolysis. It's glycolysis in reverse. And we have different enzymes for some of these steps and a tight level of regulation to avoid the cell falling into this loop where it might break glucose down into 2-pyruvates and take those 2-pyruvates and make glucose and then break that glucose down into 2-pyruvates. We don't want that to happen. That's just going to burn energy needlessly. So these processes are compartmentalized and regulated through different signaling molecules and through different enzymes. The one that we really talk the most about is PFK, or phosphofructokinase, the enzyme that catalyzes step 3. This is one of the key regulatory enzymes in glycolysis. It's controlled by feedback inhibition and allosteric regulation. And as we saw, ATP can bind to this enzyme and turn it down. ADP or AMP can bind to it and turn it up. Citrate can bind to it and turn it down. This is a key regulatory molecule here, allowing the cell to be very sensitive to what energy does it have on hand right now and what should it be doing with the glucose that it just consumed and began to metabolize. And again, ATP, ADP, AMP, even citrate all act as signaling molecules to keep this process running smoothly. And all of this allows the cell to regulate glycolysis and gluconeogenesis specifically, but also balance its energy production and its consumption. Glycogen synthesis and glycogen breakdown. Synthesis is being catalyzed by the enzyme glycogen synthase, breakdown by the enzyme glycogen phosphorylase. Both of those are going to be regulated primarily by glucose 6-phosphate levels, which activates or inhibits specific enzymes based on cellular needs. When glucose 6-phosphate levels are high and sugar is in excess, glycogen synthase is going to be turned on, allowing the cell to make more glycogen and store that excess sugar for later. As glucose 6-phosphate levels fall, that falling glucose 6-phosphate is going to be a signal for glycogen phosphorylase to turn on. Well, if glucose 6-phosphate levels are low, that means glucose is low, so we might as well start breaking glucose off of glycogen and releasing it to the body. Animals store glycogen in 2 primary places: selfishly in muscle cells, where muscles store that glucose for their own needs, and selflessly by liver cells, where the liver is storing glycogen on behalf of the entire body. Both of these stored forms of glucose allow for the cell, muscle cell, or the body to have quick access to energy. And then we have fats that are stored in adipocytes for longer term reserves. But remember, even though I've been saying seasonal, and that's true, we even mean long periods of fasting, because we begin to mobilize and metabolize fats even while we're sleeping. Plants can do very much the same thing. They're making sugars through photosynthesis. They're hydrating carbons every time the sun is out and they have access to carbon dioxide and water. And oftentimes plants can make more glucose than they need. When they do, they can store that excess glucose in their own polymer called starch. Starch is a heterogeneous polymer. It's a combination of amylose, a coil of glucose monomers linked together, and amylopectin, which is very much like glycogen in our cells. So plants store this excess glucose as a glucose polysaccharide as starch, and that provides the cell with its own short-term energy reserves. Plants can also make and store fats for longer-term reserves as well, and we find plants storing fats and starches in seeds primarily for their own developing embryo. And finally, metabolic pathways are interconnected with intermediates shared across all of these processes, and this allows the integration of metabolism, the intersection of these many pathways, But to make all of this make sense and be feasible, we require efficient and effective regulation. These enzymes have to be controlled by signaling molecules to ensure that these metabolic pathways are coordinated and we don't start getting into these wasteful loops that I mentioned with gluconeogenesis and glycolysis, and we don't let very valuable energy molecules go to waste either without being metabolized. And that's it for this lecture. So, as always, let me know if you have any questions on this material. This is it for our week on glucose metabolism. And then next week, we'll go right into the electron transport chain and oxidative phosphorylation.